Early in the discovery of this molecule and the mechanism by which it reproduces, there were some questions about exactly what went on. And there were three models that were proposed. The first one was the semi-conservative model, which is what Watson and Crick tended to believe in and support. This was the idea that an entire strand of DNA would be conserved when DNA was replicated, and that it would then be paired up with an entire strand of new DNA. This was called the semi-conservative model because one strand of the original DNA would be conserved, and there'd be one strand of new DNA. In contrast, other biologists proposed a conservative model in which the entire original strand of DNA, both sides of it, were conserved. They thought that each piece acted as a template. The new strands were formed, but then those new strands came together, creating a completely new length of DNA, while the original strands of DNA came back together. This was the conservative model because the entire original piece of DNA was conserved. Others proposed a dispersive model where bits and pieces of both strands of DNA combined and came together so that you got two jumbles that collectively made identical strands of DNA, but each one had pieces of both sides. Ultimately, it was determined that the semi-conservative model was the correct one, but it took an interesting experiment to make this apparent. Two molecular biologists, Meselson and Stahl, decided to test the semi-conservative model. Now, they understood that bacteria, which form DNA, are able to take up nucleotide precursors. What I mean by that is the pieces that come together to make DNA. So, for example, cytosine, guanine, adenine, and so on, and the chemicals that are formed into those pieces. All these precursors are nitrogen-based. That is, all the base pairs, all four of them, have a great amount of nitrogen in them. And so Meselson and Stahl grew these bacteria in a medium where all the nitrogen was made with heavy isotopes. An isotope is just another version of an atom which has an extra neutron so that it can weigh slightly more. So these bacteria were in a vat, growing, making new DNA, reproducing, and they're picking up this heavy nitrogen isotope. So after several generations, all of the DNA in these new generations of bacteria had the nitrogen isotope that was heavy, and so all of their DNA was heavier than the DNA of a typical normal or wild bit of bacteria. Then they transferred these bacteria to a medium with lighter nitrogen, back to the normal kind, not the isotope. And the idea was that now they could track how much nitrogen was being taken up and where, so that these bacteria, which were completely saturated with the heavy nitrogen, would now begin to make new DNA with the lighter nitrogen. And this way, by examining the DNA, they could tell which bits of DNA were from the original strand, which had the heavier nitrogen, and which ones were from the new strands, which had the lighter nitrogen. So after one generation in the lighter nitrogen, they took a sample of bacteria. They isolated the DNA, ran it through a centrifuge, and saw where it settled in. After a second generation, they took another sample, and they kept doing this until they had samples from several different generations of bacteria. So this diagram by the Lady of Hats is in the public domain. She created this to show people how this experiment came out. So over here we've got bacteria that have used the heavy nitrogen, N15, to produce their DNA. And then they were given a medium of N14, the lighter nitrogen. And here's what happened. Initially, 100% of the bacteria had the density of the original heavy nitrogen. After 20 generations, 100% of the bacteria carried DNA that was of a medium density. It wasn't as dense as the original nitrogen, but it wasn't as light as the lighter nitrogen. It was right in the middle. And then a curious thing happened. Every 20 generations, the amount of this intermediate DNA was split in half. This suggested that each time one strand of DNA was being replaced with the lighter nitrogen, while one strand of DNA had the original nitrogen. Likewise, the amount of bacteria with completely new light DNA increased each time by about 25%. This experiment confirmed the semi-conservative model because as the bacteria took up the lighter DNA, half of their DNA contained the heavy stuff, half their DNA contained the lighter material, and so they all had DNA with the lighter material. As the bacteria reproduced, they began to create strains that had just the lighter nitrogen in their DNA, but the original bacteria kept the heavy amount. As the generations progressed, there was a greater percentage of DNA made from lighter strands because all the new DNA was being made from the lighter strands, and there was no replacement of the N15 strands.